We use our in-house Chinese AD cohort, and we were able to identify new AD risk genetic factors. My students use deep machine learning technique in order to uncover the nonlinear interactions among the genetic risk factors. And we came up with a genetic risk prediction of AD as shown on the right part of the slide. So we have the lifetime genetic risk scoring system for AD prediction. Our study actually was one of the very first deep learning models for understanding the AD genetic polygenetic risk. And it allowed the development of genetic biomarker tests for AD risk prediction, diagnosis, and also subtype stratification. Now, that was the genetic risk prediction. Now, I want to talk about other types of biomarkers, not the genetic biomarkers, but the protein biomarkers. Now, with the brain imaging, such as the amyloid PET scan, this is now the golden standard for AD diagnosis. There's also the MRI. There's also the biomarkers in the cerebrospinal fluid, which measure the protein biomarkers in the CSF. And these protein biomarkers that I listed, they are the so-called pathological protein biomarkers. Now, these tests, they are often either expensive or there's a need for special equipment. I understand in Hong Kong, there are only 2 amyloid PET scans, or they are invasive, such as the CSF extraction. So we believe that there's an urgent need to come up with blood-based biomarkers. Now we know that the pathological proteins, such as amyloid beta, such as tau, they are altered in the blood of the AD patients. They're simple, they're convenient, and they are not invasive. However, we believe that we can do much better than this. We want to come up with a simple blood test for early AD and also very accurate. So again, it's work done by my PhD student, and it is made possible because of the technological advances. We made use of the technological breakthroughs that allow us to measure blood protein biomarkers that are at low level. So it allows us to measure more than 1,100 proteins in a very small volume of blood samples. So we made use of this technology, the ultra-sensitive protein detection technology, and combined it with high-throughput data analysis. And we were able to overcome the bottleneck of traditional blood protein detection.